Hi. Yeah, so I'll be going back more into kind of what's some of our bread and butter for some of us. And in short, before I dive into it, I think about the game theory concepts and try to root them in the biology as a mathematician and a physicist, now looking at cancer research. So next slide, please. Oh, shit. I love this. Okay. We do lab meetings and it's like, I'm like so used to saying next slide, please. I can do it myself. All right, okay. Just before I dive into the math and the science, in Cleveland, we work on ancestral lands. This is not just a past issue, this is the current oppressive state of the United States, and it currently affects people who are alive today. This is also to say that like Cleveland is almost 50% African-American, and this is not something that is reflected yet in our communities in science, and we have a long way to go. But I think it's important to recognize this as we move through science and think of this as an active process where we can all be doing better. Okay, so you've all seen this figure from Jake earlier today. I've included like, you know, the number of papers that I could include would be like 5 papers from everyone in this room individually. But to talk about how basically we are aware that cells interact both on a experimental level, more recently using these cool game assays that we have in our lab, but more broadly, there's been a ton of work already on looking at how these cells interact and using both Lotka-Volterra methods and game theory methods sorry, I'm just going to stay here for a second. It's the problem when I have control, is that the fidgeting is not as successful. So there's a lot of evidence that these cells interact. There's a lot of anecdotal evidence. You know, most biologists accept that cells interact, but part of the reasons we look at it is so that we can have the mathematical tools and the mathematical descriptions so that we can take these very precise control methods that our math allows us to have and apply them. So this here is the replicator equation. We've already seen it a couple of times, W being the fitness. And in 2-player games, we're very often a bit obsessed with this little quadrants, because basically the relative sizes of this B minus D from the C minus A from the payoff matrix basically determine the outcomes that we can have. So they're basically just, if we know those, we know what the outcomes will be. So the stuff that I've been thinking about is this ABCD. Traditionally in game theory, we think of them as basically just 4 independent numbers. But we know in biology, these things are not independent. They're not independent from each other. They're not independent from concentration as we think about how these games change. So one way of thinking about it is that you've got the growth rate of your 2 types. So your 2 players, this might be sensitive and resistant or wild type and resistant. We move quite quickly to W and R. We can think of it as, you know, those off-diagonal terms are the growth rate in the presence of the other. In the presence, it's the payoff, but this in biology, we think of this as the growth rate in the presence of the other type. So we can think of these as actually the potential of modifying that initial amount. So essentially, if there's no interactions, what we don't expect is a 0 off diagonal. We expect a constant row, because basically the growth rate doesn't matter how many of each type you have. And actually, we can think about those in terms of concentration as well. So the cool thing about this, at least to me, maybe to some of you here, is that those growth rates are not, that concentration dependent growth rate isn't something we don't know anything about either. We look at drug response curves all the time. Again, these is kind of trying to unite areas of study here. And so we know about things like the Hill coefficients and the Hill functions, which aren't, don't get me wrong, they're not perfect. They don't cover every situation, but they're one of our models. They're one of the things that we do see sometimes for certain types. So you might have, in this case, there is a trade-off between resistance and growth rate, but you might have an increasing concentration on the x-axis, and your wild type will die off quicker than your resistant type, for example. So what this means is that there's actually, in our game space, this doesn't correspond to being at the center and not moving. It corresponds to moving across the game space. So if there's no interactions, we'd still be moving in a diagonal line, basically minus what x equals y. That depends on the difference between those two curves at each point. So that you'll cross at the center when those two growth rates equal is the point where you'll intersect on your drug response curves. So now we want to be thinking as mathematical oncologists, mathematical biologists thinking about game spaces, we don't want to be thinking about the null expectation being at the center, or just as we change concentration, the expectation should be, if in this case, this very traditional idea of what happens with drug concentration, we would be going down this straight line. So then the question is, if we can do that without even thinking about any interactions, what happens when we have some simple types of interaction between our cells? So now when we do have interactions, there's two different ones that I've thought about. And one is just to add a constant, and one is to have a growth rate dependent constant. So one is, a constant interaction for the cells. This can differ between the two. And one is growth rate dependent. So this could be an uptake of a factor, for example, and that's the beta and the alpha here. And obviously we can get, these are sort of our mathematical concepts, but if we take our growth rates as being those two Hill coefficient, Hill curves that we saw before, with just some random alphas, we can end up with these wonderfully wacky nonlinear curves in random areas of game space that just depend on those alphas. And with our betas, we end up with a transition. So if you have a constant difference, you basically move, translate across the space, this straight line that you'd expect with no interactions. And our alphas, we get, because of these Hill curves are nonlinear, we get these nonlinear effects in that trajectory in game space. So another way, again, this is just taking more different alphas and just looking at specific points rather than the more trajectories, but we can just see here, we can basically with these alphas, and here it was just 0.5, so it affects the growth rate by negative or positive 50%, which isn't too crazy. We can see that we can get all the way across our different quadrants and transition between those just with a very, very, very oversimplified model of these interactions, which is just that they affect it by 50% of the growth rate that you'd expect from the drug response curves as the logistic curves. So do we get this in experiment? What do we see in experiment? So the nice thing about having this interactions from our payoff matrix is that we can actually find out where, we can say, given it's this constant term, given it's beta, we can find this beta from experimental value. So anytime we have a payoff matrix, we can find it. And so Dageen, who's sitting over there, has done amazing sets of work on these game assays. And this is just one of the experiments between a BRAF mutant and a PC9 ancestral strain in non-small cell lung cancer. And essentially we can take those diagonals of payoff matrix, which we expect loosely to follow the drug dose response. And that's what we have on the left-hand side here, is that those diagonals are following our traditional drug response curves over concentration. And then we can see what those betas are just based on the payoff matrices that we're finding across concentration. And what we see here is that the beta for the mutant, the BRAF effect, is actually pretty constant and very positive across that concentration gradient. And here we have a changing value for beta. And so it might not, basically if you have a constant, it's the idea that that model is good. If it's changing, it's saying, well, actually it's not a constant, therefore you might need a different model. This is showing that here for the BRAF, beta mutant effect, this is a good model, just adding a constant, but would need to do something else, like maybe the alpha, maybe we'd have combinations now, but we can see, basically we can have kind of loosely inspired ideas for these interactions that are inspired on, based on mechanisms of potential interactions. So obviously this is one specific model, basically we can take the future, this is very new work. So, but the future of here is we can take these equations, we can get analytical expressions for our game coordinates over concentration. We could take a ton of different models. We could be looking at radiation response and having like thinking about those interactions, because anytime you have basically a growth dependent form for your cell, you can basically then easily, with this way of thinking, have a form of your interaction, and you can look at whether that matches with experiment. Yeah, so then that's kind of, I've raced through it, but that's what I'm thinking about. If anyone's, it's very new stuff in terms of, you know, bringing this to like preprints. It's not up yet but this is the things that I've been building the other work on, you know, when do interactions modify, how do they modify what we'd expect in an experiment? This is kind of the bit that fills in the, you know, science isn't as linear as we might like to sometimes portray it as, but this stuff kind of fills in that gap, and hopefully it can be useful in interpreting games so far. This is our lab. There are more people. Jake's lab is huge. But yeah, just thank you to everyone there, and thank you for listening. Yeah.
Thanks, Rowan. That was cool. I guess I'm intrigued about just thinking about this conceptually.
Yeah.
You're saying that the drug or whatever the dose is going to modify the interaction.
So the idea is that the interactions, the way we define a payoff matrix, is to say there's a growth rate in one and other. And this isn't actually even having a drug-dependent interaction term. There's two, here it's just that the growth rate, the intrinsic growth rate is being, the two models are, one is that the intrinsic growth rate itself are just modified by the drug concentration, and the modification is either a constant beta or constant relative to that intrinsic drug-dependent change. And even that just creates these like non-linear effects.
So, I mean, it's a simple assumption, but has massive impact.
Yeah, exactly. The idea is like, it's super simple. We haven't even come into like alpha and beta being, like we do see here that basically like that beta for the BRAF isn't drug dependent at all, but we do see some drug dependence here. Like that's just a constant added, like independent of the intrinsic growth rate. But here, for example, we have like it changing, and it might be that we can reflect that with an alpha or, you know, a simple model. But yeah, it's like, these are so simple and they're still getting.
I mean, but I guess the reason why I asked for clarification is because it has a big implication. Because obviously there's many people building games of and ignoring these potential interactions that might be just swept under the carpet because they never thought about it that way.
Yeah, exactly. It's like even what we're seeing is effectively like even very simple interactions can move you like massively in this space. And we know, you know, the different quadrants have huge impact on our results. So it's like even extremely simple interaction models can give us a hugely different games, and therefore hugely different outcomes. So it's like, we don't need super complex things, and we can relate game theory to, if we relate it to the biologist better, like this kind of understanding of a modification and an expectation, then we have a better language to discuss what's going on.
Very cool.
Thank you.